with, that they can grow on. But of course, sometimes it's not so easy to understand how they achieve this. And this is what I want to elucidate with my research. I also want to apply this knowledge to create then improved organisms with synthetic biology. And this could be bacteria that degrade pollutants or that produce valuable molecules, but for example, also plants with new properties of, for example, higher crop yields that provide food advantages to the population. And of course, I cannot achieve such ambitious goals on my own. Therefore, I'm very interested to collaborate here in Leiden in the Faculty of Science, for example, among others, with plant biologists to work on understanding and engineering plants, with computational biologists to get a better understanding of metabolic regulation on a computer, with bioprocess engineers to plan efficient biological processes, and with ecologists to understand how bacteria live together with other organisms and perhaps influence each other. So with that being said, in the frame of this format, I'm also, or everybody is also asked to put their research in the context of more fundamental or more applied research and how this research could contribute to society in the short or in the long term. And well, how does my research contribute to basic science, to an understanding? Well, I think hopefully all of you are aware that microbes are really ubiquitous. They live everywhere. They live in our gut. They live in the coldest habitats in the Antarctic. They live in the hottest volcanic springs on the planet. They can make a living everywhere and form the basis of all ecosystems. And I want to understand how they can do this, because I believe that characterizing the metabolism of microbes means understanding their success and their lifestyle and their interactions with other organisms. And for a more applied perspective, I think that new enzymes, which I have discovered and hopefully will also discover, may lead to applications in biotech, and that engineered bacteria and plants are, of course, crucial components of future sustainability strategies. So with that being said, I would place my personal research somewhere in the bottom right quadrant of this scheme here. I think it's definitely more a fundamental kind of research. It helps us to understand why microbes make a living the way they do. And I think it can definitely have an impact on society, as I will show you in a few minutes. But I think it takes a bit longer until this impact realizes, maybe 10 years, maybe 15 years, because this basic work that I do needs to be backed up and followed up by more applied studies to realize the full potential of discoveries. As I said, in my case, really basic curiosity-driven research questions in my postdoc in Germany have led me to new discoveries and applications. And that is what I want to share with you in the next 5 to 10 minutes. And then I briefly want to mention how this ties into the research I want to do here in Leiden as an assistant professor. So basically, my postdoctoral work in Germany started with a very simple question. And this question is, how can marine bacteria in the ocean make a living? How do they get their carbon? Because, well, it's just water. There's not much in there. How can they make a living there? And I was going through old literature to try to find a starting point to my research and found hints that a partly characterized metabolic pathway, a way for the bacteria to get their carbon, might be relevant. This has the rather complicated name beta-hydroxyaspartate cycle, or BHAC for the rest of the presentation in short. So I looked into this. I found one gene in genome databases that might be a relevant part of this pathway. And that was the start of this exciting journey of discovery, because I found out that this gene is part of a gene cluster of 5 genes. And to understand what they do, what about this metabolic pathway is relevant, I then purified these 5 these 5 proteins to really study their role in detail in the laboratory. And to make a long story short, I could show step by step the functions of 3 different of these enzymes and how they form a linear pathway. The biochemical details are not so important here. I just would like you to focus on the big picture. So I was stuck with these 3 enzymes and this linear pathway for quite a while until I could understand what the fourth gene in this cluster is doing, marked in red here, DHCD. And this actually was so difficult because I found out then that it accepts a metabolite as a substrate that is completely unstable. You see here this metabolite called iminosuccinate has a half-life of just 1.5 seconds. So that means it decays, it goes away really quickly in solution. And this enzyme does an incredible job at picking up this very unstable metabolite and converting it into a more stable metabolite. With that being said, I had figured out the complete reaction sequence of this pathway, the beta-hydroxyaspartate cycle. And in the process, I had found a completely new enzyme, the founding member of a novel enzyme family of primary imine reductases. And actually now I'm also looking into how this enzyme class could be applied in biotech. And this also means that this pathway was no longer linear anymore. It's a cyclic pathway, as became clear. And as its summary, it converts two molecules that are called glyoxylate into one molecule that's called oxaloacetate. But as I said, the details are not so important here. But what I want you to remember is it takes two molecules with two carbon molecules each, two carbon atoms each, sorry. So C2 plus C2 equals C4. That means that these smaller molecules are put together into one bigger molecule with 4 carbon atoms. That will be relevant for the rest of the talk. And after figuring out the gene cluster and the reaction sequence of this metabolic pathway, the BHAC, me and my colleagues went on to characterize this from the smallest scale, from a scale of nanometers or micrometers, where we determined the crystal structure of the beta-hydroxyaspartate cycle enzymes, onto a larger scale of centimeters or meters in the laboratory, where we looked at the algal secretion of a metabolite that the bacteria then make a living with using this metabolic pathway, the beta-hydroxyaspartate cycle. And not only we looked at this in the laboratory, we also went on to do field work on the relevance of this metabolic pathway. So we went to islands in the North Sea and sampled the bacterial communities there to understand how they get these metabolites from the algae. So that was done on a scale of kilometers, basically, and going into an even larger scale of thousands of kilometers, I looked in the relevance of this pathway in global metagenomes, so genomes that colleagues had basically sampled from each part of the ocean, the Atlantic, the Pacific Ocean, and I was very excited to find out that this pathway indeed plays a role in microbial communities all over the world. So with that being said, with the characterization of the beta-hydroxyaspartate cycle from the smallest scale to the biggest scale, I would like to switch gears a little bit and focus then on metabolic engineering with this pathway. And here again, it's very relevant that we have in mind that this pathway converts C2 molecules into C4 molecules. So there are also other pathways that follow a similar pattern, a similar metabolic route. But these pathways follow the formula 2 plus 2 equals 3 plus 1. And why is this important? Well, because this C1 is a molecule of carbon dioxide, which gets released in the metabolic process. And as you all know, carbon dioxide is a climate gas, and the more of it is released by bacteria, which are everywhere on this planet, the more of an impact it can have on the climate, basically. In contrast, this very simple equation of 2 plus 2 equals 4 shows that bacteria are able to do an efficient assimilation of these C2 compounds using the beta-hydroxyaspartate cycle. And so with that being said, with the beta-hydroxyaspartate cycle as a very efficient pathway for C2 assimilation and also for photorespiration, a process in plants which is linked to photosynthesis and which also deals with the conversion of C2 compounds, it now became relevant and very interesting to engineer different bacteria for efficient assimilation of C2 compounds and to engineer plants or algae with a more efficient photorespiration pathway, which should help them to do more efficient photosynthesis. And the time is short. I would just like to give you a very brief overview of how this can be relevant for bacteria that should degrade plastic. One of the most abundant plastics is PET, or polyethylene terephthalate, which is made up of the monomers terephthalate and ethylene glycol. And here it's important to know that ethylene glycol is actually a C2 molecule, and therefore it can be fed into this pathway, this metabolic route that I'm talking about the whole time, to be assimilated following the 2 + 2 = 4 formula. And therefore we took bacteria, Pseudomonas putida to be specific, and we introduced the beta-hydroxyaspartate cycle genes into these organisms. Then I set about to do directed evolution with these engineered bacteria, which is a powerful approach to improve enzymes or organisms. It was recently honored with a Nobel Prize and will have a big impact on biotechnology. And also in my case, this directed evolution showed some promising results. As you can see here, the evolved strain with the beta-hydroxyaspartate cycle could grow 50% faster on ethylene glycol, this compound of PET, of plastic, and it could grow 25% more efficient, as you see in the red bar here. So this is very promising as one part of a strategy to use microbes for the degradation and bioremediation of PET plastic. And switching gears again, I very briefly want to talk about how this pathway can be used for more efficient photosynthesis in plants. For that, we teamed up with experts in Düsseldorf who used the beta-hydroxyaspartate cycle genes for the metabolic engineering of Arabidopsis, a model plant with this pathway. And so they chose a mutant that normally requires a high CO2 atmosphere for growth. We could show together that the enzymes of this pathway were localized to the peroxisomes in the plants where they should be, and that in a low CO2 atmosphere, there was a clear phenotype, a beneficial effect. So you see here on the left, this mutant strain of plants grows very badly at a low CO2 atmosphere, but with this pathway, with the beta-hydroxyaspartate cycle, it grows quite a bit better under these stressful low CO2 conditions. And therefore, we think we're really on a good way to use this pathway to improve photosynthesis in plants. So with that being said, I hope I could show you in a very brief overview that this discovery and curiosity-based research that then led to the characterization of a novel metabolic pathway now can have very exciting applications in the bacterial degradation of plastic and the engineering of plants for more efficient photosynthesis. So what do I want to do with this? How do I want to move on in Leiden here? For one, I want to try to identify new biocatalysts from different bacteria from everywhere in the world in a very efficient way by screening in a high throughput manner and using fluorescent proteins as shown here as a reporter. So I want to set up such a screening workflow. And also I want to continue to work with marine bacteria and study how they could be engineered efficiently to clean up pollutants or to maybe improve aquaculture systems, which are becoming more and more important. And I'm looking forward to do this with my team in the next years here. So with that being said, I'm at the end of my talk already, and I'd like to discuss with you a very general question. I hope I could convince you that synthetic biology does have great potential to tackle sustainability issues. But what do you think, what is the main obstacle in realizing the potential of such exciting technologies? Thanks for your attention, and I'm looking forward to the discussion.
Thank you so much, Lennart.